get to listen to me twice in less than 24 hours. Okay, so kind of picking up with this theme of Paul, I mean, you know, you want to maybe focus a little bit on the things that can only be uniquely done with the very high intensities that are available at the FELs at these very short wavelengths. And so, just to remind you, which maybe is like beating a dead dog again. Well, you know, it's inspired so many people around the world. I would have to say I have to beat it a little bit. So, but remember the idea that we spent some time on was that you have this big molecule and you want to know its structure to atomic resolution. And it might have 3 million atoms in it. And in principle, you can do this by this technique of X-ray coherent interactive imaging onto the. But of course, there are all these analytical questions that you'd like to answer because, as I told you in the first lecture, a lot of things are happening to the electrons. You know, they're moving all over into different configurations during the course of the pulse. And the image that you measure on these detectors is, of course, the integral of everything that you have. So, you know, it's kind of a tricky problem. And in this lecture, I want to go through as to how you would actually, you know, researchers are making steps toward this. I just want to go through the steps that people are doing. Before I do that, of course, you know, there's a big difference between crystallography and non-periodic imaging. In crystallography, you get an n-squared enhancement for the number of unit cells that you have in your crystal. You get all these Bragg peaks, and you can reconstruct what you have. And so one of the very first applications, the very first beam times at LCLS was this, you know, this landmark experiment of Henry Chapman and this very large set of collaborators in, again, Pierre's least favorite journal, right? But
and our journal has much, our record is 6,000.
Oh, really?
Yeah.
Oh my God.
I need to.
Yeah.
Right, and it may even be that some people in this room are on this, are on because it actually took a lot of AMO techniques that were used to actually do this experiment, right? And so the experiment was on this photosystem 1. It's a membrane protein with a molecular weight of megadalton. They had to take like 3 million diffraction patterns, but they had these really tiny crystals. And you know, they looked at the near field and the far field scattering, and it was at a rather long wavelength. It was at like 2 kilovolts, and so the resolution they got was only in this like 8 angstrom type regime. So it's not really what they wanted to get to, but you know, it's an incredible first step. And so they did this, and they were able to match the synchrotron structure at this very low resolution. But it demonstrated the whole idea that you could do this sort of, you know, crystallography on very small crystals, right? Of course, you know, the real idea was not to have crystals at all. But this has spawned, you know, I didn't make a list of all of them, but it has spawned so many different implementations of this over the last 5 years that is quite remarkable. And even in this Swiss light source, the drug companies in Switzerland want to fund a station just dedicated to this crystal light. So I mean, it's a winner. It's definitely a winner, right? Now let's go to the other thing that, you know, that first image was on, right? And these were published back to back in Nature. It has also a huge number of authors, and many of the same authors, in fact. And it also, you know, it uses AMO technique and injected its, you know, particle. In this case, it was the Mimi virus. It's the largest known virus. It has almost a micron in size, doesn't crystallize, kind of large for cryo microscopy, etc. And they got single-shot scattering patterns and reconstructed in 2D, like 30 nanometer type resolution. And of course, it, you know, but 2D is not 3D. And 3D is something that they realized only just recently using this data, right? These many diffraction patterns. is they actually reconstructed some shape of the giant meaty particle virus, now published in a very reputable journal with slightly fewer authors. But out of that huge slew of diffraction patterns, they got around a couple hundred, and they used an orientation recovery algorithm, which is tricky, obviously, since it took so many years, and reconstructed, at this 100 nanometer type level. But it's still pretty far from what people, it might be nice to get to the few angstrom level, right? So it's a little far. So how are we going to get there? Okay, so this was a nice viewpoint that was written from that PRL. And this is by Keith Nugent, who's quite a well-known person in coherent diffractive imaging. He says, okay, well, there are still open questions on the impact of electronic damage. That's kind of what we study. On X-ray scattering on femtosecond time scales. And so this is again motivating us to continue, even though it seems really messy and dirty and all that other kind of stuff. But it motivates us to continue along what we're doing, right? And so in this lecture, what I want to tell you about is some studies done by Argonne and his group on X-ray interactions with isolated molecules. This is a nice step up from atoms. And they get quite a bit of insight from experiment, but I want to point out before I start a little bit is that there isn't actually a full theory that describes what happens in this very intense X-ray field. And then I want to talk about something slightly larger that actually has a theoretical component associated with it, and that is an experiment by Nora Vera and her collaborators, where the MD simulations work quite well on this rather well-defined molecule. And the theory there was done by Robin Sanford and his group at CFEL. And then what I want to tell you about is that we want to move on. I mean, C60 is nice, but it's hardly a biomolecule. You want to move on to something slightly larger. And so what we're moving on to are these X-ray interactions with clusters. It's kind of a step toward this direction. And there are some new tools that we can get to. One of them is that there's a new tool that allows one to define the interaction strength on your sample, which is kind of important if you want to model it, by doing coincident spectroscopy and scattering in a single shot. And then I want to tell you about the new tool that's available such that you have an X-ray, you have your big pump, and then you have another probe that can come in and look at, kind of what happened. And then tell you just very briefly that there was a surprise in the first experiment on that X-ray using that technique. These are all very recently developed in the last year or so. And then finally, how we would hope to be able to advance in computation that will take us toward this 3D imaging in clusters.
Linda,
Yeah.
you referred to damage to these biological molecules by these X-ray.
Yeah.
By that, I think you still refer to these ionization processes, right?
Well, it's both. It's both this sort of electronic, like where are the electrons are going, and when you get to the large biological molecules, you'll see what happens is that you tend to make a nanoplasma because first you have a lot of ionization, lots of electrons leave. But then you have, kind of a charged
Space
thing.
charge, yeah.
There's a charged thing, and so that traps, some of the electrons. Some of them come from Augers and are kind of slow. They get trapped, right? And so now you have this kind of weird background of these loose electrons, not necessarily. So it's just a tricky problem.
Yeah. So I'm trying to understand in the context of, for instance, work which, for instance, Leon Sanche did more than a decade ago, where they showed that actually it's not necessarily the radiation which causes the damage to these biological molecules, but at the secondary ionization events.
Yes,
the recolliding.
yes. Well, what's
electrons.
interesting about that is, some of those secondary ionization events that may happen at a slightly longer time scale.
Right.
And for the diffractive imaging, the only things that actually show up in your image are those that are during pulse. Right. So many of those things will be like not visible.
Right.
I mean, they will. So what's really good about this coincident spectroscopy and scattering is that you get both the long time dynamics and the short time, from the scattering. And so because you get the long time dynamics, you have a hope that, you can model what happened with all these secondaries, as well as, what happened on the time during which the strong X-ray pulse was present. Okay. All right, so this is the paper I want to talk about. I think it's a really nice paper. It was led by Artem Rudenko, and it talks about ultra-fast charge rearrangement and nuclear dynamics on this strong X-ray pulse on a small polyatomic molecule. The question, of course, is just how does this microscopic response of the small polyatomic system compare to an atom? Okay, it's going to be somewhat different. There are more degrees of freedom. And can we understand the competition between the charge rearrangement, column explosion, photoionization, and all these other things in a small molecule that actually tends to mimic a biomolecule because you have a heavy atom and then you have the lighter atoms around it. And so Artem and his gang chose this nice methyl subinol molecule. So it has a selenium here. The selenium is a Z equals 34, and he could compare directly to a krypton, which is a Z equals 36. They took this rather fast LCLS pulse and focused it onto there. And in their ion spectrometer, they were able not only to measure the charge state distribution, but also the ions in coincidence with each other and get the momenta. So from the momenta, of course, it's very good because you can get the kinetic energy release and you can get a lot of dynamics from that. Okay, so what they first did was, okay, let's just look at the charge state distribution and compare with those two things that have very similar Z. Because, that's where all the absorption happens on, most of the absorption happens on the heavy atoms. And so what they found is if you take a look at the charge yield, the yield is a function of charge, what you find actually is that the krypton charges here that go out to 16, that go out to charge state 16, are very similar to the total methyl selenide charge, which includes not only the charge state of the selenium, but also that of the carbon, and assumes that even if you don't capture all the protons, that the protons are ionized. And so you immediately know from this, right? Even if the absorption has happened on the heavy atom, that the charges aren't localized there. It's distributed, and something else is happening. But this is not all the information that can be gotten because they have this nice recoil ion momentum spectrum. So what you can do with that is you can look at all these different channels and see how the kinetic energy released in this explosion compare to how they would be if they were at the equilibrium position. Okay, and so that's what is shown in this slide for a number of different channels. So there's the selenium 1, carbon 1 channel. And it breaks up into all these different channels. The selenium 2 plus, carbon 2 plus, selenium 5 plus, carbon 2 plus. And this would be where there's not as much charge migration from one place to another. Okay. So if you take a look, these sticks down here represent the kinetic energy. Okay, so what this is the ion kinetic energy for these different channels. And these sticks represent the kinetic energy release from that equilibrium position. So what you can see is that while it works pretty well for this channel, selenium 1 plus, carbon 1 plus, when you get out to these other channels, these equilibrium kinetic energies, for example, let's see here, take a look at this one. Here's selenium 2 plus, carbon 2 plus. This is what you observed, but over here is sort of what you predict. And so it's less than the prediction. So what does that mean? It means that, the thing has expanded or done something before this explosion, right? And so, yeah.
Could you explain? I mean, just take the top curve. It says selenium, but then the dots are selenium, carbon, single charged, and the circles are selenium double charged, carbon double. So
So this is the kinetic energy of the selenium fragment.
the selenium fragment includes the carbons.
And this is what you would expect
They're not
from the
single
channel.
selenium.
No. Well, this is this channel, selenium 1 plus carbon 1 plus, right? And then the red is this other channel, selenium 2 plus carbon 2 plus. And so what you see, right, is for this low charge channel that it matches more or less the equilibrium. But when you get to some of these other charge states, it doesn't match, right?
So I'm confused. The vertical lines are what?
The ion kinetic energy for the selenium atom in this explosion calculated using a Coulomb explosion at equilibrium geometry, which for selenium and carbon.
And they seem to be all the same value, is that right?
Well,
The vertical.
you see there are different scales here because carbon versus selenium, right? So in the Coulomb explosion, it's slightly different kinetic energies that you would get for the two. And let's see. Right, and so what?
Oh,
So, okay. Let's see. So if you take
I
a
see.
look,
No,
you see what I'm saying?
Yeah.
So here's selenium 2 plus 2 plus. So in the equilibrium geometry, you'd expect to peak around here. And you actually get it for this 2 plus channel a little bit
I
earlier.
see.
You see what I'm saying?
I understand.
And then for the
Yeah.
selenium, let's see.
So it's the position of the peak
Here's
that
the
should
5,
look
right,
at.
position of the peak here. The 5 plus 2 plus channel, you'd expect something out here that exploded from the equilibrium position.
Okay.
But what you see is something down here.
That's it.
So something has happened, right? You can see it right from the data. And if you plot this data instead as the product of the selenium, and carbon charges and the sum kinetic energy for these calculations, assuming the Coulomb explosion at equilibrium geometry, you see this, right? But then, okay, and so you see most of the data for these different channels is below the equilibrium geometry, suggesting that, on a rather fast time scale, something has exploded. And if you actually plot what's shown here is, let's say you assume that the molecule is stretched out to 3 angstroms instead of being like at 2 angstroms initially. Then what you see is this dashed line here. And this dashed line here, more or less mimics most of the data that has been seen. So, the kind of conclusion that you have is, oh, well, this selenium carbon bond, which started at 2 angstroms, has gone out to like 3 angstroms before there's this charge, there's this stretching on the same time scale as the explosion. Right? And then there are these other things, these other interesting channels that are going above this Coulomb explosion simulation at the equilibrium channel over here, right? And so what's going on with those, right? So some you can explain by the fact that there's an ultra-fast dissociation before the Coulomb explosion, and others you can't. So if you look at the yields for all these different channels, for let's say the 1 plus 1 plus is a 1 photon absorption, and then there's some mixed in and 2 photon, 3, and so on and so forth. You see that the yield goes sort of as the intensity to the nth power for an n photon transition. But then, if you take a look at those oddball things, what's happened is that those oddball situations where the carbon charge is higher, the carbon charge should be lower than the selenium charge because it's initially, at the initial stage, because the selenium is charged and the carbon is less charged, right? But if there's some charge transfer going on between the two, then something can happen. So you see when you have this state where these triangles, where the carbon is highly charged and selenium is less charged, it falls along the same kind of line as this very highly charged selenium and carbon 3 plus, indicating, oh, well, maybe you started out in that configuration, there was a charge transfer going on. Okay, and so here is the idea. These slides come from Argonne. And the idea is that, okay, you started out with this 3 plus 5 plus type stage. But then there's this very ultra-fast charge redistribution before the Coulomb explosion, such that you end up with this 3+1+ stage that seemed to fly in the face of the kinetic energy release. And so the idea here is that the charge rearrangement by some kind of interatomic molecular Auger decay most likely happens before these protons are moved far away. And so, and the typical proton velocities are very fast if you have some kinetic energy. So the point is there's a lot of complex dynamics going on, but maybe I'm wrong, because maybe there's someone out here in the audience who's working on this problem, maybe from, who knows, a time-dependent Schrodinger equation, who knows, some approach. But it's basically a challenge for theory. Can one really predict this behavior from, first principles? It's a very complex behavior, right? And it's of importance if you want to look at the bioimaging, because of course if the nuclei are moving on the same time scale as the electrons in the pulse duration, it affects whatever you see in your image. So it's a pretty tricky issue. And then I want to tell you, yeah.
Is anything known about the potential surfaces? Do you know if anything is known at all?
I personally
Or is
don't
it
know.
such as there's barriers
Brett
or-
is here from Kansas State. I mean, does Artem talk to you about this?
Well, I don't do quantum chemistry, so
Yes, I know.
he's not going to talk to me about those potential surfaces. But
No, but
I mean,
just in
quantum chemists
general.
can calculate anything, right? So
Well, that's what they say, but it takes a little bit. It probably takes a little bit of persuasion. Well, I know it takes a lot of persuasion to get anyone to do any particular molecule. It's a little bit like maybe there should be an experiment first, or hand in hand. Sometimes it.
No, I mean, so the basic questions, for instance, whether there's a barrier-less or barrier-type reactions going on here or not.
Yeah. I would say I
Yeah.
don't know the answer to that quite. But there are a lot of theorists here in the audience.
I'm one of them, but I don't know.
Yeah. Okay, so the next thing I wanted to tell you about was this femtosecond X-ray induced emission C60 at extreme intensity. All these experiments were done at the LCLS. This was an experiment led by Nora Vera. And here, there was a more close coupling between the experiment and the theory. And so there were a lot of things that could be learned from this. And it's a very simple thing. There's an LCLS pulse, it's about 500 eV. They had a number of pulse durations that were sort of nominal. This also goes to show that I think the metrology in the laser labs is a lot better than the metrology at the FELs at that point in time. But normally, they tell you, oh, well, this is your pulse duration, but in fact, you had to calibrate it to figure out what the pulse duration was by putting in, an atomic gas. And they had, various high pulse energies onto this C60 molecule. And then what they measured using a magnetic bottle spectrometer were the fragment ion yields and, ion kinetic energies of each ion, but not necessarily in coincidence with each other. But still, that gives you a lot of information. Okay, so the general picture is that you have this pulse, this is singulated, of 30 femtoseconds. And, and this left side is on this very small spatial scale of 10 angstrom far here. And so you see your fullerene here. And, initially you have electrons just coming out. But then, even by the time you've gotten to, the middle of this pulse, which is, fairly energetic pulse, the buckyball has disintegrated and the electrons are coming out. If you go over to the larger spatial scale, you can see at that exact same time, the electrons are everywhere, right? And then by the end of the pulse, of course, the ions have caught up and they've exploded everywhere too. But you really want to, in order to model this particular experiment, you have to follow everything through all the way out to the long time scales, because it's not just that femtosecond pulse. Okay, so from the modeling, you can extract what's going on in high, mid, and low fluence situations. And so, because for fluences that are larger than this, the yield is primarily carbon atoms. The modeling was basically done all on carbon atoms and their energies. So what you can see is that in the short pulse case, you have these ion charge states that go down. In the intermediate pulse case, that go out a little farther in pulse duration, and in the long pulse case, it goes out into higher charge states. And the modeling starts off atomistically, and then once these ions and electrons get out into the continuum, it becomes classical again. And so, it's something that I think is tractable. It's tractable on a larger scale. And so what you can see from this is that the difference between a C60 and a carbon atom, right, directly. So here's the carbon atom in green. And what you see is this alternation of 1 to odd and even alternation that we saw, that you always see because you have the photoelectron and the ocean. So every photon that you get in gives you, 2 charges. So you can have odd. But then, when you look at the C60, you can see you have this large abundance of charge state 1. Large abundance of charge state 1 is due to the fact that you can have, or is modeled because you can have molecular Auger effects that give you 2 C+, C1 plus atoms from adjacent neutral atoms. And then what you can also see is that in the atom, you have more high charge states because you don't have recombination like you can have in the C60. Okay, so now, beyond charge state yields, you can also see these frustrated absorption X-ray transparency effects both in the atoms and the C60. This is for 90 femtoseconds and 4 femtoseconds, and you can do the ratio of the two. And what you can see for the atomic versus the C60 is that relative to the atom, you have a suppression of the high charge states due to the electron trapping and recombination. But if you do the ratios, it looks like it's a very large effect also. And then, if you do, they did this very nice ionization as a function of fluids, both for the atomic case and for the C60 case. And what you can see, in the atomic case, it's very simple. You have photoionization, you have your Auger, and then you end up with a final charge state as a function of fluids. And for the C60, you have not only those two things, the photoionization and the red Auger, but you also have the secondary ionization. And then that will give you a different total ionization, but not only do you have the secondary ionization, you also have the recombination, and so then you end up with this purple for final charge state. Okay, so then when you model things, so when you model things, okay, it started out with this initial radius about 3.5 angstroms, and then you can see for a 90 femtosecond pulse at very high fluids that you can obtain, that you can have, a pretty large expansion, from down here all the way up to here by the center of that pulse. So, it's pretty, well, it's dramatic. You really need to show pulses, right? And then you can see that the charge state continues to evolve, and this is something that's required for, these modeling, all the way out on 2 time scales of picoseconds. Okay, so it's of course, there are other things in there. There are the chemical bonds, and the interesting thing is now we can look at these very short pulse durations, right? That are maybe more relevant to energy. The interesting thing is that the effect of chemical bonds on the kinetic energy and charge states almost vanishes when you get to the very short pulse duration. So this is like a good thing for the molecular energy. So this on the left is plotted the average atom kinetic energy difference as a function of the fluid. So you want to look out in this high fluids region. What you can see is like it's almost zero difference when you get out into the high fluids. And similarly, the difference for the charge state distributions for full model versus bonds is small-ish, right? Okay, and then, they're very thorough in their modeling, right? So you have to keep adding. You add one thing, add another thing. So this is what was done. So they took the difference between the model